Hello, everyone. This is Mr. Baker. We are starting Unit 3. In this unit, we are going to focus on DNA, its function, and what it's used for, and as it acts as a way for organisms to get traits. And that's going to be our big push as next unit and the unit afterwards. We're going to talk about those traits a little bit more and how they're passed down. So this is kind of a foundational unit. We're also going to talk about the history of how DNA was discovered, right? So as we're getting going here today, we're going to talk about how DNA is genetic information and how it's stored. So looking at the history of DNA discovery, right? We've talked about how cells were discovered and how scientists were getting better and better at looking inside cells. In 1869, Friedrich Miescher actually was able to isolate the DNA molecule from a cell. He was able to get the DNA out of it. He didn't call it DNA, though. He called it nuclein. About 12 years later, Albrecht Kossel actually named deoxyribonucleic acid. He confirmed what Friedrich Miescher had been doing, but took it a step farther in that he saw that there were 4 different nitrogenous bases, 4 different nucleotides, as we're going to call them now. And he came up with the idea, the name of deoxyribonucleic acid. In the late 1800s, though, a lot of science starts focusing on war efforts as World War I starts. And it's going to continue to focus less on biology and inheritance the same way until closer to World War II. 1944, Oswald Avery talks about DNA as the transforming principle in cells. That means that the DNA itself is what is causing cells to be different from one another and to do different things from one another. And then Chargaff is going to work in the same time period to say pretty much the same thing, but he links it to inheritance as well. So not just that cells are different because of their DNA, but that DNA is what lets the cells of one organism give rise to the traits in their offspring. It wasn't until 1953, though, that we got a good idea of what DNA's chemical structure looked like, what that molecular structure entailed, right? And it is really a story of 3 scientists. Rosalind Franklin was working on studying the structure of molecules using radiocrystallography, while Watson and Crick were trying to actually surmise the structure of DNA. And they were doing that from a mathematical approach, while she was doing imagery. Right? Someone got a hold of a picture she had taken in the lab of DNA, and that's the picture you see in the bottom left that looks like a circle with an X. That is a kind of top-down view of DNA, and it's called Photo 51. That person gives it to Watson and Crick and says, hey, I found this image, this photo of the radiocrystallography done on DNA. I thought it might help you. They were down to about 3 different possible structures for DNA, including the double helix. This kind of gives them a jump start to go straight to double helical structure. They prove it through math that we're not going to get into, and they publish. The same journal that they publish in, Rosalind Franklin publishes her work done through imagery on the structure of DNA. This being the 1950s, however, she is a female, they are males, they are published first in reading order, and she's published second in reading order, which means that to the general public and the scientists who are reading these, it looks like Watson and Crick have done the work and Rosalind Franklin has confirmed it. But they were completely different and they were studying different things. They were not working together. Now, jumping to modern science, we understand that they all should get credit for the discovery of DNA's helix structure, and we can give them the credit they deserve, not just giving Watson and Crick the accolades. Last unit, we talked about nucleic acids, right? This should be in your notes. Please notice the 2.5 in the top left-hand corner, right? This is the exact slide even, right? We have this single nucleotide because the monomer of nucleic acids are nitrogenous bases right here. So we've got this guanine here. I'm just reminding you of this, all right? Guanine is one of the four, right? Chargaff, because he's doing a lot of work with DNA, one of the things he posits in the time period he was working are a set of rules. And we now call them Chargaff's rules. But effectively, he said that the amount of adenine will always be the same as the amount of thymine in a DNA segment. And the amount of cytosine is going to be equal to the amount of guanine in that same segment. And if you put them all together, you would get 100%. These are what I call base pairing. It's what a lot of scientists now kind of call them, or just base pairs. It is the rule that describes the structure of nucleic acids and how they bond together to form DNA. Right? So you can see here in the ladder, we've got C's and G's always paired up to one another. We'll talk about this more in class, and we'll even do some modeling. For now, though, I want you to try to apply this math, right? So hit pause, read through these questions, give me the two numbers, and I'll look at them in your notes when I grade them. When we take DNA and we see it in cells, we are usually seeing chromosomes. Right? Chromosomes are the condensed strands of DNA. DNA coils in itself, but your cells, all cells really, wrap those coils around some proteins called histones that help to coil them together tighter and tighter. The coils, all right, are going to supercoil together into chromosomes. If you've ever taken your hoodie string or a shoelace and you've twisted it until it spins up into a coil, this is a very similar idea to that, except for we've got some histones in the mix to make the initial coils. As we've talked about today, we've looked at the history, we've looked at the structure, and we've looked just a little bit at the function. We're going to keep talking about the function in this unit. For now, make sure you've got everything in your notes. I also want you to add, this is the last thing, I want you to add here at the end a sketch of DNA in a ladder model with 5 nucleotides on each side. Make sure they are linked up A to T, C to G. There was one in the notes, so you can go back and just draw that one if you want, or you can come up with your own. I'm going to check for those and the answer to the question when I check your notes. I'll see you in class.